locally stress applied to the bacteria, and then the alternative nitrogen source. Then the third one is optimizing operational parameters, which is used for like scaling up the production, because usually this is very important for the industry to know the strategies about like to scale up the production of the MK-7, the vitamin K, I mean. Okay, so basically vitamin K, what is vitamin K? I think you already know, right? So basically it is fat-soluble vitamin. It's also known as the clotting vitamin. Why? Because it has the ability to do the blood clotting in our body. Then there are basically three vitamin K kinds of vitamin K. Yeah. So the first one is vitamin K1, which is derived from the phytoquinone, which is called phytoquinone, derived from the plants. And then the second one is the vitamin K2, which is also known as menaquinone, and then this is also derived from mainly the bacteria. And the third one is vitamin K3 or menadian. This is the vitamin K that usually found in the supplements or like in the other products. Okay, so basically the vitamin K have like this chemistry from formula, which is this is the ring, and then have like side isoprene chain, and this length is like determining which vitamin K we want to extract or it's detected. Okay, and then I will specifically focus on the MK-7 or MK-7 because it is specifically produced by the bacteria, but usually in like low concentration, because it's just like mainly from their biosynthesis so it's part of their metabolic pathways. So what's the benefits? Why it's important? So MK-7 have like several benefits. Usually these benefits also like benefited the elderly, because I currently live in like East Asian, which the population of the elderly is like increasing every year. So they should like maintain their health by consuming something that can maintain their health. And then vitamin K is basically one of them. So there are three main benefits that vitamin K can offer, which are enhanced vitamin D3 induction of osteocalcin, which is very important for the bone health. And then the second one, prevent vascular calcification. This is very important for the heart. And then the third one, to improve the insulin resistance, which is usually the diabetes type 2 patients will suffer. Because of these benefits, currently vitamin K is in high demand. So the researchers will try to like increase the production of vitamin K. Okay. Oh yeah. Actually, why vitamin K2? Because the bioavailability is high in MK7 in vitamin K2, because usually body will like more readily available for like our but our body absorb the nutrition. Okay, then so did you guys recognize this? Yeah, this is the natto, or like fermented soybean food from Japan, in which they made this by fermenting the soybean with the Bacillus subtilis, and then after they are fermented, the researchers try to extract the Bacillus subtilis that already adapt with such condition,
As
nutrient condition.
considerable.
Yes. So, uh, so basically, soybean rich in protein, right? So, uh, usually the bacteria will like adapt with the uh, depends of the environment. So, if uh, and then the researchers will try to obtain the Bacillus subtilis, which they name it again as Bacillus subtilis subspecies natto. And this is the strain that most commonly found when we culture the Bacillus subtilis natto. Actually, I forgot to take one of my picture because actually I feel like it's kind of beautiful, but uh, it is very troublesome if we want to count the bacteria because we need to uh, you know time time management have like good time management about that. So basically Bacillus subtilis natto is the Bacillus subtilis derived from natto, the fermented soybean, and then it is also considered as gram-positive bacteria. It is also a spore-forming bacteria and biofilm-forming bacteria. So it can also synthesize MK-7 as part of its metabolic pathways. So biofilm-forming bacteria is like they tend to live together. Like so in natto, as you can see, there is like a slimy texture. Yeah, it is because of the natural of the Bacillus subtilis natto to form the sticky slime texture. Okay, so let's move on to the MK-7 pathways. So there are basically some pathways, which the main one is these two, the MEP pathway, as you can see in the figure. So the MEP pathway is related to the isoprene side chain. So here they have like several enzymes that are have important role to form the vitamin K structure. So for the MEP one is a isoprene related to the isoprene side chain, and then the MK1, or usually we call it as sikimate or sikimate acid pathway, which this pathway is important for the naphthalene ring, the ring that I've mentioned before. So there are basically have like main structure, right? So the first one is the ring, and then the second one is the isoprene side chain, and then this ring and side chain will be joined by this enzyme called MAN-A. Usually this enzyme will join this, and then they will form the vitamin K. Okay, so basically the studies will either increase the cell proliferation, or uh, like have some control with the enzyme expressions. Okay, so how they can enhance the MK-7? There are basically recent advance have like three mains, which are strain improvement, which include the mutagenesis and selection, then adaptive evolution with chemical modulators, and the second one is by metabolic engineering. This is the I want to mention, uh, which includes the overexpression of key enzymes and precursor supplementation. And the third one is fermentation, scaling, like the first one is biofilm based fermentation, and then basically it's for like scaling up the production. Okay. Oh, wait. Okay. So biofilm is the structured community of microorganisms that adhere to the surface, which they are will uh, embed with the EPS, extracellular polymeric substance. And this matrix were contained of polysaccharides, proteins, and DNA. Then, what's the purpose? The purpose are to protect from the environmental stresses, antibiotics, and the host immune system. Basically, this biofilm forming activity is like to make the bacteria feel comfortable with the environment. So they are feel comfortable and they feel like comfortable and safe. Yeah, safe from the outer attacks. Yeah, so basically this is the bacteria communicating with each other to form the biofilm. Then yeah, this is the mechanism of the biofilm forming, which the cells will attach first. After they attach, they will do the proliferation, and then they will produce the EPS. Then after that, usually there are like several attacks, right? So the outer like cells will release first, because usually the attacks, and then they will just release first, and then the cells under, like the cells that attach to the surface will maintain their location, so they can proliferate more and more. So the fermentation strategies includes like there are three main, which are the batch fermentation, chemostat fermentation, and the fed batch fermentation. So basically, if it's inside the bioreactor, they have like propeller, and then the propeller will spin according to the speed that we can adjust. Then for the first one is the batch fermentation, which is considered as closed system, and the nutrients were introduced at the initial process, and the final product will be harvested in the end. Then the second one is the chemostat or continuous fermentation, which is considered as open system. Why? Because the nutrient inflow can be equal to the products and residual nutrient outflow, which is continuously feed. So basically, it's continuously feeding the bacteria to produce more whatever they produce. And then the third one is the fed batch, which is also considered as the open system, but they in this fermentation they don't remove the products. They don't remove the products and just add more nutrients. Yeah, just like that. And then this is the objectives of this today's presentation, which we would like to review how the strategies can enhance the biosynthesis of vitamin K productions. So this first study titled "The Effects of Alkali Stress of the Growth and MK-7 Metabolism of Bacillus subtilis natto." So this paper used the Bacillus subtilis natto as the main strain, and then they will do the batch and dynamic fermentation, which is they will shake the flask to enhance the bacteria proliferation, and then they will apply the alkali stress, which is they will apply pH 8.5 to the bacteria. So like they want to see how the stress can affect their metabolic pathways. So basically, they will first prepare the seed culture, and then they will adjust the mediums to ferment the bacteria to do the fermentation, and then they will uh, they prepare the pH, the range of pH from the acid to alkaline. Okay, so this is like several figure that shows how the pH can affect the growth. So basically, if you see in the first picture, the highest production of like living bacteria produced is in range of pH 5.5, which is in this case means that this pH is like preferable by the bacteria to grow. And then after that they will increase the pH until like 7.5, 8. They show like decreasing some decreasing trends, and then after they continue until like 8.5 to 9, there is like increasing trend, and which is this usually some bacteria have like adaptation with the condition. Like for example, this pH condition, it's a bit extreme, right? From 5 to 8.5, because usually bacteria will start to form like spores or something, and then to in order to protect them. And then after the they see this trend, they continue to check the MK-7 concentration which produced by the bacteria that is adapt with the specific pH, which they use the 8.0 and 8.5 until 9, and then they will compare to the controlled group, in which the bacteria first will be adapted by the pH. Then they after that they will see the changes. So, and basically after we saw the second figure, the highest is from the bacteria adapted by the 8.5 environment, in which this means that the bacteria have like comfortable with that 8.5 environment, pH 8.5, sorry. Okay, so then they continue to see how the 8.5 pH can actually change some like some parameters. So for example, for the number of living bacteria, actually the pH, the alkaline pH can increase the number of living bacteria. And then, but as you can see, usually hello, yes. Yes, bro. Okay, so I will continue. In this case, they found some like some of the parameters are increased, but for the sporulation rate were decreased. Wow, this is actually very interesting phenomenon, because actually the viewers, if the spores are like few amounts, which this can indicate that more cells can stay in a favorable environment, in which we can conclude that the pH 8.5 can be one of the favorable environment for the bacteria. Okay, then the others like the C1 like OD 600 is the biomass, and then the glycerol concentration. Oh yeah, I forgot to mention that they use the carbon source here. The carbon source was the glycerol, not the glucose. Yeah, usually they use the glycerol to grow the bacteria, uh, to grow the Bacillus subtilis natobacteria. And then for how about the menacanin-7 concentration? So for the alkali stress group, like after several hours, so they try to observe the 24 hours until 72 hours, and then they found that the alkali stress can like continuously produce the MK-7 or vitamin K. So means that this alkali stress uh, can increase the production of this vitamin. Oh, and then they will like have to study more about the pathways. So the pathways will be shown by this table, which are there are like some enzymes in the to produce the MK-7. Say previously I have mentioned about the pathways, right? And then they have like the enzymes there, so we can see like it's expressed. Usually we want the main enzymes of MK-7 forming uh to increase. Okay, and this is the genes that related to the enhance of the glycerol metabolism. In this case, because very it is very important, because usually they need more energy to produce something. So these genes, if it's rising, like it's over regulated, yeah, up regulated, so that means it can we actually wants it to be affecting the production of whatever we want, whatever product we want. So usually this one is like the main parameters of the production of whatever, usually like in bioethanol, and then other bacteria secretion. And then have you remember that I mentioned the main enzymes? So these main enzymes are really important for the MK-7 production, because if we regulate their metabolic systems, yes, so we can see like if we regulate these main enzymes, so basically uh, we can increase the MK-7 productions. Okay, so the summary of these studies were the alkali stress can activate the MK-7 biosynthesis pathways, like actually it's increase their pathways, their enzymes pathways. Okay, so now we can move to the second paper, which titled soy protein hydrolysate, or usually we refer as SPH, induce menacanin-7 biosynthesis by enhancing the biofilm formation of Bacillus subtilis natto. So I have I have mentioned before that biofilm is really important for the this MK7 because actually I forgot to mention in the introduction because the vitamin K is like tightly bound in the cell walls. So it means that we should proliferate the cells to in order to increase the MK7 production. And then in this paper, they will use the Bacillus subtilis natto, same strain, and then they have like strategy. First one is the batch, and then static and dynamic. Then they combine this fermentation strategy. So they call it as two-stage aeration, and then they will use the soy protein hydrolysis as nitrogen source. Actually, SPH is considered as like how to say more expensive than the soya peptone, but because of their uh tiny peptide, yeah, so means that it is easily digest by the bacteria, so they will produce more. Okay, and then this is the two aeration stage fermentation. So the first one is focusing the first stage is focusing the to produce the biomass. Then for the second stage, usually the oxygen will be reduced, and then they will be focusing on like producing whatever they produce, whatever we want to get, like whatever we target. So, but in this case, this figure I got from the study from the ethanol production. So yeah, basically they do the same thing, but their goal were different. Okay. So then for the methodology, they basically prepare the soy protein hydrolysis. They have like different hydrolysis time from 4 until 24, and then they activate the Bacillus subtilis, and then they ferment, which they use so how many parameters? Yeah, actually three, because one is the like static, and then the second one is dynamic. Then they combine these two strategies into one called two-stage aeration fermentation. Okay. So for the second paper, they have like the figures, which the first one they show the result of the submerged fermentation, in which the SPH can increase the biomass significantly. But however, the MK7 remains stable or even decreased from the control or soy peptone group. So basically, they are comparing between the soy peptone that we usually use to culture the Bacillus subtilis natto, and then they compare with the soy protein hydrolysis. Then which this showed that the high biomass can suppress the MK7 biosynthesis because they prioritize the growth, the cell proliferation in this stage, because this stage is like static, so they will just not static. This one is dynamic, which is they usually shake the flask to proliferate more the cells. And then for the second figure, show the result of biofilm based fermentation, which is they just left it just as it is. So it means like it is static, right? Static fermentation, which in all groups showed increasing trend of MK7 content. Also they also attach the real picture, as you can see, and this picture is showed how the biofilm will form during that dynamic fermentation. And then how about if they combine the stage the stages into like the two aeration strategy. So as you can see in the picture, they show the increasing trend with the two aeration stage, which combine these two, static and dynamic. And then it means that after the cell proliferation, like after they grow massively, then they will start to produce secreted more of the vitamin K. Okay, so let's go to the next figure. So the next figure showed the early stage of like 48 hours. So in like SPH media, the spore fraction, the spores in spores like in red area, the red area, spore fraction is lower than the soy peptone, in which in this case the bacteria in vegetative state longer, because the SPH can provide the accessible nutrient. Okay, then for the late stage, in like around 100 hour, the SPH media spore content can be increased because the cells use the nutrient quickly, so the bacteria can be tired and trigger the transition to spores a bit early than the previous one. So means that in SPH media, the faster nutrient uptake, and they can also enhance the biofilm structure. So that means they can quickly produce the MK-7 production, and also quick to produce the spores. Okay, so that is it. Oh yeah, I forgot to click it. Okay, so the next one, how about if they compare from each SPH with the different time treatment. So basically, if the longer treatment of the SPH can reduce the size of the peptide, so they can easily digested by the bacteria. So for the first one, showed the increasing trend of all of these parameters, including the biofilm EPS, and then the biofilm protein, and then the biofilm gamma PGA. This is actually the biofilm related parameters, in which the increasing the SPH hydrolysis time can promote the biomass production of the bacteria, and then can also promote the addition, and it means that the biofilm will be very like they will produce much of the biofilm, and then they can also produce the protein such as the TSA and BSIA, which is this protein is really important for stabilizing the biofilm. And then gamma PGA usually used for the nutrient exchange parameter, which is longer the hydrolysis time, they can facilitate the nutrient exchange more. So the metabolic will be faster. Okay, so what's the impact of the hydrolysis time with the biofilm genes. As I mentioned before, there are like some enzymes that they have to express this because usually this genes expression are related to the metabolic pathways. So there are some of the genes are decreased because these genes is for the regulation of usually spore, which is if the spore is like decreasing, so means that vegetative cells or active cells will produce can produce more. And then for the SINR genes, usually it is expressed as the repressor of biofilmetric genes. Then the others one that I highlight with the green arrow increased, which in PSIA is the protein genes that related to the biofilm surface stability, and then the test A and EPS or extra polymeric substance that I've mentioned before, to maintain the structure matrix component of the biofilm forming during the biofilm forming. Okay, which because the quorum sensing or quorum sensing, how to say, it's the bacteria communication. If it's down regulated, they can also encourage the biofilm matrix formation. Okay, then as we can see here, also the genes related to the MK-7 or vitamin K production, they have like some of genes are downregulated because especially in Mandy and ROA, as I mentioned with the red arrow, which is these two is related to the naptokinase ring, the ring that I mentioned before in the chemical structure of MK-7, which this can be suppressed, but because naptokinase ring is just like this ring, but usually the most important one is the isoprene side chain, because this chains is like how to say, they can relate with the bioavailability. So this is usually will be the focus one to upregulate the genes that related to the isoprene side chains, because as we know, MK-7, vitamin K2 or MK-7. So MK-7 means like they have like 7 isoprene side chains, and which is this is the chain, the exact chain to be easily digested by our body. So it is actually the most important for this gene upregulation. Yeah. So means that the SPH, the longer time of hydrolysis, can also enhance the MK-7 production by the activating the isoprene pathways. Okay, so what happened? What happened when the hydrolysis time was longer? Were longer because if longer hydrolysis time can also lower their molecular weight or MW, because lower molecular weight can also like the bacteria can easily digest and produce more of their secretion, and then actually this is also important because hydrolysis time, which when we increase the time, which is the energy also high, right? So if it's applied to the industry, usually they won't take the longer time, so they will start to adjust the time, like to optimize, sorry, the to optimize the time, so they won't uh produce like more energy, because more energy means like more expensive, so expensive product people don't want. Okay, so that is like the key of this production. Okay, so yeah, basically small peptides under 1 kilodalton will be easier to be absorbed by the cells or the bacteria. So they can like, if you say, how to say, if for example, like if you eat rice with you eat porridge, it will be faster if you eat porridge, right? Yeah, so it's basically like that, but yeah, in more specifically in peptides. Okay, so because the researchers already have like in like more lab scale uh, lab scale studies, so I will move on to like the, how to say, it is also lab scale, but more like what happened in industry, but in like mini lab scale. Yeah, what can I describe it? Okay, so this third paper uh titled as Sustainable MK-7 Production Through Continuous Fermentation in Biofilm Bioreactors. In this case, they use the bioreactors, but the lab scale one, to like mimic the one that usually used by the industry. So why they use the bioreactors? Because they want to scale up to faster the process, to make the process faster, and then to maintain the controlled condition. Okay, so in this case, absolutely they will use the Bacillus subtilis natto as the main strain, and then for the fermentation strategy they use the continuous. So did you remember the continuous one? Yeah, the continuous is means that they will continuously feed, and then they will continuously let out of the cells. So yeah, so it will depends on the like retention time. I will explain later. So basically it is a dynamic fermentation, of course, because in bioreactor, as I've mentioned before, they have like a propeller, so they will spin, and then we can adjust its speed. And yeah, usually we will adjust according to like any bacteria characteristics, because yeah, usually some of the bacteria such as Bacillus subtilis natto, usually we tend to do it a bit faster, because they, if not, they will like form the biofilm. Yeah, this also like the challenge of the industry, because biofilm is actually really disturbing, because the biofilm can increase the maintenance fee, like cost, the maintenance cost. And then they use the bioreactor, and then they add the plastic composite support, or PCS. This plastic composite support is to support the cells to attach with the propeller. So the cells will like produce more and more whatever they secrete. Okay, then they will optimize the operational parameters. In this case, it's very important for the industry, because usually they want they want to use as less as possible of energy. Yeah, so to maintain like the so it's not it's not really expensive. Like if more energy, so more expensive. Okay, so remember the key. Basically they will do something like this. Yeah, as I mentioned before, the Pirater have like have this propeller, and then this propeller, they set up the plastic composite support that already coated, how to say, yeah, coated, or they made it, they will cast as like uh, from various agriculture result. Like usually they will use soybean waste, yeah, because soybean, as we know, is rich with the protein, right? So they use the soybean meal to make this like plastic composite support. Also also it will be uh how to say, it will be supported by the plastic because yeah, it's more rigid, right? So basically the bioreactor will be designed, they design the bioreactor so design as like to attach the composite support, and this is for cell adhesion, to support the cell adhesion. And then this cell adhesion obviously can promote the biofilm formation, can which they hope that it can enhance the bacteria secretion. So also it is high advantageous, especially for the biofilm forming bacteria growth, because yeah, if they are biofilm forming, usually they will tend to attach with the surface. Okay, so they want to make them comfortable. Mm. So if they feel comfortable, they can produce. So it's just like if you are doing your thesis, and then you thesis or scriptsy, so you will you will tend to have like to search for like comfortable place, and then that 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 time you will start to produce your scriptsy. Okay, so weird analogy, but yeah, that's okay. Oh, actually, in our lab, we already do something like this, but it is for the bioethanol production. Yeah, so uh, they usually use this to ferment yeast and to produce the bioethanol, the second generation, or the 2G bioethanol. Mm-hmm. And then hopefully I can continue. Haha, lol. Okay, so at first they will arrange the plastic composite supports, or PCS. So as you can see, they use the soybean hulls as like this is waste, so means that they hope it will be cheaper. Like in production, it will be more cost effective. Mm. And then they will start to develop the biofilm first. So they will start to like deep immerse the support, and then they will make sure that the cells were there, and then they start to ferment, which is like into parts. So first one is the batch fermentation, which is just like not the continuous one, which is, yeah, we just add some nutrients and then that's all. But the continuous fermentation, usually we need to adjust the nutrient inflow. Okay. Yes, and then this is the results of their study, which shows the how the MK7 and glucose production can affected uh, by the by their design of the PCS and then attached with the bioreactor. Okay, so the figure A, as you can see, A and F. So figure A, at the start of the continuous fermentation, the MK7 level. So you can MK7 level is like in the blue one, and then the glucose concentration is in the orange line one. Okay, so why I compare this one, like especially I highlight these two results because the A one have like high a low flow rate and dilution rate, but the retention time is 120 hours. And then for the F1 have like a high flow rate and then dilution also high, then the retention time is lower, like around 12 hours. And you can see that the line actually almost touching each other from the A figure and the F figure. They separate from each other. So I will explain first from the A. So the figure A, so at the start of the continuous fermentation, the MK7 level can be dropped, but then after a while they stay steady. They stay steady, and then during this phase, if I can zoom a bit, yeah, the glucose was around like 90 grams per liter, which shows that this glucose also play important role to the MK7 production. And then they fed the system, right? The flow rate is around 12.5 milliliters per hour, which is the MK7, which can be a bit lower because the the glucose is not enough for there. So basically, with I forgot to explain how the flow rate and dilution, also the retention time. So basically, if you have like a cup of milk, yeah, so the milk is the flow rate is like the speed when you pour the milk inside, and then the dilution rate is like the speed how you like fill up the the cup, because this the the pouring speed and the usually it's like in line, right, with the how speed you fill up the cup. But then the retention time is when it is hard, but yeah, retention time is like how the milk can stay before you let it out, before you pour out the milk, like because retention time is like how the medium is inside the bioreactor before they will start to flush out. Okay, so means that if retention usually the retention time will be affected, will affect the biofilm forming, because usually biofilm forming have like longer retention time is better because they will attach more. But in this case, in the A, so because the fat system is just like 12.5 mil per hour, which is very low of the glucose intake. So maybe this because there is no enough of raw material of like glucose can slow down the MK-7 process. Okay, and then for the F1, the final test, they use the 125 milliliter per hour, which is 10-fold higher than the A treatment. They also start I mean like among all of these flow rates they test, the feeding rate of this faster, like because they are 10 folds faster, give the lowest MK-7 production. Why? Because the bacteria didn't have enough time to make the MK-7 before the liquid got flushed out from the bioreactor, which means the A1, the problem is just nutrient intake, nutrient uptake, and then the F1, the problem is the time. So yeah, very confusing actually, but this is why optimization is very important. So they tried several flow rates and dilution rate, and with also different retention time. So like lower flow rate, they use the high retention time. Then the fast flow rate, they will use the low retention time. So that means these two parameters were like really can be considered for this MK-7 production. And then in this all, the author choose the E, the figure E, because they said the result was like more stable. And then yeah, as you can see in the picture, actually they have like very good figure like for the optimization. And then what we can say from this figure is lower rates can ensure enough nutrients and retention time for the biofilm formation and MK-7 synthesis. But this higher rate can we can explore, see how far the nutrient supply and retention time could be pushed for this bioreactor fermentation. And then, but it is important to see if it's too high dilution rate, can the productivity of whatever we target, we because they have like low retention time, so they have no enough time to synthesize whatever they produce. And then actually this is very important for the MK-7 vitamin K production, because we will be focusing in more like developing the biofilm, right? So this can be like very concerning. The challenge for the scale up production. And then after that, they calculate the production, the productivity versus the dilution rate of those treatments, which is they compile into one figure, which shows that when the dilution rate increased, the MK7 productivity also went increased, which means that adjusting the fermentation time can help um, make more of MK7 production. So for example, in this figure, the productivity increased from the 0.12 to 0.33, which is as you can see the earlier line, and then usually they have uh, they have like more nutrient intake. Okay, then and then after that they will in 0.42, yeah, the highest, the peak one, and then after that uh drop dramatically to after they increase the dilation rate, because as I've mentioned before, the retention time is very important to make the biofilm, and then this case, because biofilm is very important for MK7, so means that there is not enough time to produce the MK7 in that dilation rate. But because it is fast, usually the the industry or the company will choose the faster, because faster means that you can produce fast but in like limited time, just a short time is better, usually is better, because yeah, we also in the industry usually they will think about the energy consuming process. Okay, so they will like they in this study they provide the information about how the operational parameters of the bioreactor can be adjusted and can be optimized to see which one or which parameters that could be adjusted to produce more of the bacteria secretion, especially in this case vitamin K. Okay, so for the summary of today's presentations, there are three mains that I will mention. The first one is the biofilm development can significantly contribute to improve the vitamin K biosynthesis by Bacillus subtilis natto, because biofilm is like we make the Bacillus subtilis comfortable enough to for them to release whatever they make inside during the metabolic system, and then they can also for the studies can also regulate the metabolic pathways, which is it is very important to maximize the MK7 production, as we know that those genes inside the when expressed like in for some example like enzymes playing important roles to synthesize, like in vitamin K they tend to like join the two chemical compound and make the vitamin K, something like that. And then also integrated fermentation approach can also provide the sustainable and scalable solution for large-scale MK7 or vitamin K2 production. Then this case is really important for the industry, because sustainable is like very important for the industry, because first one, not only they will contribute to the environment, but also they will also contribute like for the cost effective one. So means that if low energy, the environment will be happy, and then the company also will be happy because yeah, many they will have like many benefits from them. Okay, so here is the references that I use for this lecture, and then if you are curious, you can just check these references and yeah, maybe study. And then okay, thank you for listening. Yes, so this is
Okay,
my
Miss
notes.
Pini.
Yes,
Thank you very much for the nice presentation, informative and at the same time crystal clear.
yeah,
Uh, Before we continue to the discussion, let me deliver the summary of your lecture.
yes,
Uh, the production of K2 vitamin is so low, so there is some experiment is carried out to
yes,
increase the production. And then the benefit of vitamin K2 is, for example, enhancing vitamin D3 production and improve insulin resistance.
mm,
The next is the producer is a bacteria,
yes,
namely Bacillus subtilis natto, strain natto, and it is isolated from natto, Japanese fermented food.
mm,
And then you explain also about the glycolysis and EMP pathway in order to
yes,
explain the production of the K2 vitamin production. And you explain also the strategy, strategies. These
yes,
following high uh, strategies are strain development, and then metabolic engineering, and then fermentation process optimization. In this case, you explain about more about fermentation process optimization.
okay, yeah, correct.
Yeah, and then you explain also the fermentation strategies, namely batch and continue or chemostat and feed batch.
Yeah, yes,
And then you explain in detail some studies. You
mm,
review. The first is alkali stress, and then you find that the pH 8.6 is the best.
mm,
And also you review about the various pH values on the
yes,
production of K2 vitamin. And here you find the pH 8.5. And then the third is you explain about the feeding flow rate on the production of
yes,
K2. Okay. We appreciate very much for your presentation. And now we continue to the discussion section.
yes.
So now I will open the discussion, maybe session one. Please, some questions. Muhammad Abid.
Uh,
Muhammad Abid, yes, please.
Okay, okay, thank you. Okay, Miss Winnie, thank you very much for the epic materials for today. I have two questions. Sorry, before, my name is Muhammad Abid Anurido
hello,
from 22
okay,
Intake.
yes,
So I have two questions. For the first one, as you mentioned before, as first paper is about
mm,
alkali stress,
yes,
the carbon sources that they're used to fermentation is glycerol. It is not glucose.
mm,
So my question is, why not the glucose? And the second question is, you mentioned about the challenges from the industry to apply their uh, the several kind
mm,
of strategy. And you mentioned to the maybe wish the industry to when
mm,
they applied this strategy, this enhancing MK-7 strategy. So but as we know, about everything about uh, microbiology or biotechnology, especially for the modern fermentation, is not cheap.
Yeah.
uh, Everything about microbiology is not cheap. So
Yeah.
uh, if
Actually, yes.
the industry, if the
Mm.
industry applied the several kind of enhancing MK7 strategy
Mm.
that we discussed today, is it
Mm.
worth it? Like
Ah, okay.
you mentioned about all of the challenges, about all of the waste from the industry, but is it worth it for
Yeah.
industry to apply that? So that is my question. Thank you very much.
Okay. Okay, thank you a
Okay.
bit for your questions. Actually, good questions. Yeah. Lol. For the first question, why use the glycerol? Because usually glycerol is like more readily available, and then actually it is cheaper than glucose in terms of like using in fermentation. Also, because they also like have higher degree of like reduction, because bacteria tends to like readily available nutrients, which is they want something like, yeah, I don't want to chew, I just want to swallow something like that. So it's like they are really lazy. Yeah. What I can explain is just that. And then because also glycerol is actually, do you know this fact? mm, Actually it is a byproduct of the biofuel production. So that means if it's cheap, usually industry will use. So that's why the recent studies will use glycerol more. Usually it's really rare if like using glucose these days, because usually they will just use the glycerol. But there are some like uh there are some like case. So for example, my friend used the yeast, and then they use the glucose to produce the bioethanol, because they said it's better for the synthesis. But I don't really study the pathways of making the bioethanol. So what can I what can I share with you? It's just usually we will see like each character of the microorganism, and we will uh, what we will target, what target what they should produce. mm. And then for the second one, you ask for the worthy of the MK7. Because uh, I don't know if MK7 is like uh, can be easily found in Indonesia. I forgot. Yeah, because Indonesia have like younger people. Yeah, yeah, younger people population are like very uh, more than the elderly. But here in the east and also west, I think the elderly population were high. So vitamin K is like we can found almost, yeah, everywhere. But the vitamin K usually they will add the vitamin D. So vitamin K2 with vitamin D. Yeah, as I mentioned before, usually it's for bone health, and also they try to like combine these two vitamins in like one supplement, so they don't buy it separately. Okay. Uh, and then this worth why and then these vitamins, usually vitamin K2, will be produced directly from the natto. Like they should make natto, and then they extract the enzymes, something like that, and then they will extract the sticky texture of the natto, and then turn it into vitamin K supplements. But this usually they have like actually have like some challenge because the maintenance were high. So actually this fermentation strategies or something we try to develop because we would like to like uh, have something like to reduce the how to say the maintenance, the high maintenance of the if we directly extract from the natto. Okay, so just we want just the bacteria to produce, not making the natto first, then we extract. Okay, so it's actually if we already develop something like more cheap than making the natto, that means yeah, that means it is success. Then and usually the researcher will register the patent to the method so that the industry can use that. Mm-hmm.
Okay, Miss Vinny, thank you for your answer.
Yes.
The next question comes from
Okay.
Valia Almira Putri.
Okay.
Please state your question shortly and briefly, clearly. Yeah. Okay.
Okay. Thank you for the opportunity. Excuse me, let me introduce myself. My name is Fali Alimira Putri. So I have noted down a question. The question is, what are the possible side effects if someone with digestive disorders consume fermented foods like natto, a source of MK-7? Nah, and are there any solution to help the body tolerate it better, Miss? Thank you.
Okay.
So
Is that your question? Yeah. Okay.
okay, interesting question, but can you say it more like what you mean is like they directly consume the natto?
Yeah. Yeah, yes.
Okay, okay, okay.
Because yeah.
Okay, yeah, this is why the industry will have like own oh yeah, yeah. Okay, so this is why the industry will actually make this kind of supplement. So this is why sometimes people maybe don't like natto, like for the flavor itself. Not all will like it, but for me I like it. Okay, so but not all people will accept that taste. So also for the nutritional intake, as you mentioned, yeah, there is some, but this is why we try to develop how to produce the supplement without like eating the natto itself. So vitamin K2, if you found in like pharmacy, or I don't know if in Indonesia is there is available or not, but usually in the pharmacy they have like vitamin K2, and as I mentioned before, usually the vitamin K2 is from natto. So but because it's informed in supplement, I don't think it affect much like for the digestive something. Yeah, usually supplement is just supplement. You can just drink just in the served amount. The suggested amount. Yes.
Okay. Thank you. The next question comes from Ica Agnes Wulandari.
Alright, thank you for the opportunity. So good afternoon, Miss Vinny. Firstly, I would like to thanks for your informative and interesting material about MK-7. Let me introduce myself. I'm Ica Agnes Wulandari. Currently, I'm a semester 7 student of Agricultural Product Technology in the UNSRI. So I have a question, Miss, regarding your explanation.
Yes.
In the paper 2, you highlighted that the researcher used 2 stages of aeration fermentation strategy, which is at the first stage, we can call it as a normal aeration, and at And at the second stage, there is oxygen restricted. So my question is, what is the purpose of the restriction regarding of biosynthesis of MK7? Is it cannot work if we only use the normal aeration instead of use two stages of aeration fermentation strategy? Thank you.
So usually the researcher use this two stage of aeration. Oh yeah, first of all, thank you for the questions. And then, so this two stage aeration, the first stage is like to proliferate the biomass, the cells to grow massively. Usually, because high oxygen, so they tend to shake the medium, and then the culture growth. So they will shake it, and then this shaking method will increase the oxygen, and then they will start to grow rapidly during the stage, because it's just we want to have the bacteria grow massively at this stage. And then the second one is to let the bacteria it like focus more on like secreting whatever they secrete. For example, in this case, we want to the author wants the bacteria to produce the vitamin K, and in this case, they will just start to focus more on the metabolism. So which is and you said why not just the normal? It's also can it can, but this strategy can increase. So the point is we the researchers wants to search the perfect methodology, not perfect, like the method the methods to increase the MK7. And then in this case, in that paper, they use the two stage aeration. Okay.
Thank you, Miss.
Yes.
Okay, Miss Fini, I have also my question, Miss Fini,
Yes.
very basic, namely
Yes.
the product vitamin K2. Is it produced by the Bacillus subtilis natto? Is it extracellular products or intracellular products?
Yeah. That is also good question. It is actually intracellular,
Intracellular. Okay.
very tightly bound with the cells. So if we want to extract the vitamin K, should do the ethanol and methane. So usually combined between these two. But for industry, because they want it to be safe, not toxic, so they just use ethanol.
Okay, I
So
think
yeah,
uh-huh. Yes,
yeah,
please continue.
to just cells break the cells.
Mhm. I think it would be easier and cheaper when we could somehow produce somehow to engineer the product to be released from the cell to the environment. Then we don't have more step to
Yes.
extract the product to
Yes.
destroy the cell walls or membrane seal. You think
Yeah.
it is possible?
Um,
Okay.
I don't think I have seen any like that
Uh-huh.
kind of engineering, metabolic engineering, engineered strain, because as I saw, usually they will just study about the extraction strategy, which is
Mhm.
they want it to be safe and also cheap. So currently, we will use like the ethanol to extract. Yeah, just that.
Hopefully someday.
Yeah, hopefully. Oh, you give
Okay.
me idea. Thank you, bro.
Thank you. Thank you.
Thank you.
So, audience, another questions from you? We have plenty of times.
Pak Agus, I have questions.
Yes, please, Pak Budi.
Okay. Thank you very much, Pak Agus. Uh, I'm Budi Santoso, Head of
Yes.
Agricultural Technology Department. Thank you
Mm,
very much. I ever eat the natto in
mm,
Japan.
yeah.
My opinions, natto is not delicious, but
Yes. Yeah,
uh,
yeah,
Have you
yeah.
but
tasted,
it's
Prof. Budi? Have you tasted?
what?
Have you tasted?
Uh,
Where do you enjoy it in Japan or in somewhere in
workshop, workshop,
Indonesia?
workshop. I go to Japan workshop in
Ah, I
incubator.
see.
Yeah.
I see.
Uh, it's very, very many beneficial. The
Mm,
the the uh, the natto. Yeah.
yeah.
My question is, in Indonesia, so many fermented food, traditional fermented food, like tapai. Do you know tapai? Yeah.
Yes. Tapi,
Do do tapai contain vitamin K2?
okay. So that
Yeah.
is your question. Okay.
Yeah.
Tapai is basically yeast fermented, right? So it's not fermented by the bacteria. So I think the vitamin K2 will be like lower. Even like for the Bacillus subtilis, they need to have like grow in the nutrient which is rich in nitrogen source, like more protein to produce the vitamin K better because they need to be supported of like to enhance their metabolic system. So if we put the bacteria is uh like in high concentrate of the glucose but not balancing with the nitrogen source such as like protein like peptone or something. So usually the MK7 will be the MK7 or vitamin K2 will be low concentration. So this is very tricky one yeah, to produce the vitamin K2. And yeah, of course the strain usually produced by the lactobacillus or bacillus because these two have enzymes. I forgot the name, like have something, because the these enzymes also like contribute why they have the metabolic pathways related to the MK7 production.
Mm.
Yeah. So I think Popeye is not. Maybe maybe we can like explore the other
Oh,
fermented food that is rich with the protein.
okay, thank you. Bu Penny Surya, do you know tempoyak?
Tempoyak,
Tempoyak,
I don't think I ever had it.
fermented.
It is a fermented durian. Fermented
Yeah,
durian
durian,
juice.
fermented durian.
Tempoyak.
I think it's a big potential for the develop like vitamin K2 ya. Maybe
Uh,
bagus ya.
It's a possible, Professor Budi. It's possible. Possible.
Yeah.
Let's see in
No,
the future.
maybe the tempoyak doesn't have enough protein inside, so it cannot, because actually to produce the vitamin K, the raw material or the food has to be had
Robin?
a lot content of protein. Maybe we
Oh.
can find from the
Uh-huh.
peanut or another
yeah,
cereal.
peanut. Yes, that is
Peanut.
also
Oh.
Yeah,
Oh.
another cereal. Actually, our gut produce, our gut in the gastrointestinal system, they produce the vitamin K. But you see right now,
Yeah.
in the supplement
just
industry,
low
yeah,
concentration.
I know about that.
Yeah.
Supplement industry always to make a new product to become someone to become be health or something like that. But actually, we can just find from the
Yeah.
food. Mhm.
Yes.
Miss Finny, I have a personal impression
In
about the
a little
bacillus.
bit, because I just come back from the dentist. I
Yeah,
want
mantap.
Hello,
hear from
Prof.
thing. But it's good. It's good. Maybe our student next time they will find a peanut or cerealia who has a lot of content, a lot of protein, and you can like Miss Fini did in her research, ferment
Yes.
it. Maybe Pak Agus will help to do the ferment it, and we can get the vitamin K2, and then we can combine with the vitamin D or with the nano calcium. We combine and it will give advantage mm, for our what bone health. Yeah,
Bone
Fini,
health.
yeah?
Yes. Yeah.
Yeah. Okay.
Miss Finney, you
Yes.
explained that the producer of this K2 vitamin production is Bacillus
Mm.
subtilis natto.
Yeah.
I have personal impression about Bacillus is not from the good family, you know.
Mm.
But
Yeah. Yeah. Yeah.
yeah, but yeah, I know there are some strains from Bacillus that is also beneficial for the human purpose. What is
Yes.
your comments?
Uh, what is it again? I forget. What is it again?
I mean,
Sorry.
is it Bacillus subtilis the only producer
Oh,
which can or
I,
there's another
yes,
bacteria
there is.
species?
There is
Uh,
actually there are some study about Lactobacillus brevis, which is found from the yogurt, and then they found that this Lactobacillus brevis also can produce the vitamin K. So basically, it is from the Bacillus strain. Like it is the original, the nature of them, of the Bacillus strain. So, but because they are in low concentration, we want to increase, right? So yeah, usually also the researchers will use tend to use the Bacillus subtilis natto, because the Bacillus subtilis natto is like from the high nutrient, like high protein environment, so they can like adapt more in the we don't need to do anything to adapt them, and then they will adapt by themselves. So to break down the protein, and then they will produce the uh, the MK-7 by its metabolic pathways. So
Thank
usually,
you.
so actually for the metabolic pathways of them, yeah, it's natural usually. But yeah, because we want to increase, just that's the challenge.
Thank you for the explanation. Yeah. Okay, another questions, people. We have still time, but not so much. Our time is only at 12, right?
Yeah.
Another questions? Oh, Amar Farid. Okay, please state your question shortly and briefly, clearly. Yeah. Okay, please, Amar.
Thank you, sir.
5 minutes.
Good morning, everyone. I'm Muhammad Amar Farid from 2023 Agricultural Technology class. First of all, thank you for the very informative presentation, Ms. Vini. I have one question about fermentation method. What are the main advantages of continuous fermentation over conventional batch fermentation for MK-7 production? That's it. Thank you.
Okay. Thank you for your interesting question. Actually, yes. So why? Because continuous offers like nutrient inflow and outflow, right? So they can tend to like continuously refresh the inside the medium. So means that you add more every time every time we feel like the nutrient is almost almost empty, so just add more and more. So it means that also continuous will save more time, and then save more energy, because we just like keep it uh, keep it adding it, keep adding it, but because the cells will be like flush out, so they be like uh the maintenance will be lower because we already flush out those like cells or the result of the fermentation. But for the batch, because we just like one time, right? So we need to change every time. We need to change, clean something. This batch actually it's okay with the lab scale, but for the industry, it is better using the continuous because they don't want to like cleaning every time. So they will clean just in periodically, uh, if there are like some dirty standard from each company, yeah, usually they have, so they will like periodically like every week or every month, so it will be save more time and more energy.
Thank you,
Okay,
Miss Finny. Thank you. Um, I think the time is up. Again, thank you very much for the nice presentation, Miss Finny, and your time. We appreciate it very much, and we hope that in the future we can invite you again to give some guest lecture.
Okay, maybe after my publication.
We will wait. We will wait for it.
Yeah,
Let's see.
hope
Okay.
so, hope so.
Okay. Thank you very much.
Thank you.
Yeah.
Yeah.
Okay, and then I give back the time to the MC.
Okay, thank you very much, Ms. Vinny, for your insightful knowledge, and to Mr. Agus for skillfully moderating today's session. We will now proceed to the certificate handover for our guest speaker. This will be presented by the coordinator of the Agricultural Product Technology Study Program, Professor Dr. Budi Santoso, STP, MSI. To Professor Budi, the time is yours.
Okay. Thank you. Thank you, Ibu Penny Surya. Thank you for sharing your value, insight, and expertise with us. Your contribution have enriched our understanding and knowledge in the topics. We would like to present with a certificate of appreciation for your participation as a speaker in this seminar. Once again, thank you very much.
Thank you, Prof.
Thank you very much, Professor Budi, for presenting the certificate for our speaker. Before we end today's event, let us take one final documentation for the session. Dear all participants, once again, kindly turn on your cameras and take your position. For the documentation committee, please now begin taking the final group capture. With that, we have reached the end of today's International Guest Lecture. I would like to thank all speakers, faculty members, and participants for your valuable presence and active participation. I apologize for any shortcomings in carrying out my duties as the Master of Ceremony. Let us close today's event with gratitude and best wishes. Wassalamualaikum warahmatullahi wabarakatuh. Have a wonderful day, everyone.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, Prof, and everyone.
Thank
you. Terima kasih sekali lagi.
Welcome.
Terima kasih. Mudah-mudahan bisa ketemu nanti di masa
Ya,
depan.
amin.
Oke, terima kasih.
Oke, terima kasih. Thank
Ya.
you, everyone.
Thank you, Pak Agus.
Sama-sama. Saya izin leave.
Saya izin ya.
Thank you, everyone.
Thank you.
Thank you, Aurel.
Thank you ya semuanya. Ini enggak denger ya suaranya. Thank you guys. Abid makasih. Siapa lagi yang nanya tadi?
Terima kasih Bu.
Abid nyimak ya. Nyatet Abid ya.
Nyatet.
Mantap. Yes. Calon-calon penerus ini. Penerus Pak Agus.
Udah. Abid izin live ya Bu.
Oke, good job guys.